welcome to class. We're talking about metabolism today, specifically enzymes. Okay, so we saw this on Wednesday, right? Catalysts are going to speed up chemical reactions. They don't get used up by the chemical reaction, but they're going to speed it up. And enzymes are proteins that act as catalysts. Good? Okay. Whenever you're having a chemical reaction, you're expecting bonds to be made and or bonds to be broken, right? You're trying to put these things together, you're trying to pull these things apart. That is challenging sometimes. To get a reaction to start, you need something called activation energy. Often that's heat. We're going to look at it like this. So we have our beautiful reactants over here. We have our products over here. Is this reaction spontaneous? Wait, say it. I'm getting a no, I'm getting a yes too from this side of the room. Okay, people who are saying no, how come you're saying no? Because you need energy like here-ish? Okay. People who are saying yes, how come you're saying yes? Because of this thing? Okay. I can see both sides here, right? Like, whoa. We need energy up there. But the overall change in the reaction is negative delta G. Right? This is spontaneous. Okay? But it's hard to get done because of that energy bump. It's called the activation energy. You could think of something like TNT, right? Dynamite. Dynamite's gonna blow up. It gives off energy, right? It goes from really unstable to way more stable. Gives off energy in the process. That delta G is negative, right? It's a spontaneous process. But like dynamite doesn't just like boof all of a sudden. What gets it to go? Based on watching cartoons on Saturday morning, how do you make dynamite explode? You light the fuse, right? You supply, like, it needs a little bit of activation energy to go, right? Overall, it's going to get up way more, but you need some activation energy to get over the hump, right? We're good with that? There are a lot of things that you probably intuitively have similar things happening. So like, I don't know, you have a, I don't know, Pickle of the Month club that you have subscribed to. It's beautiful. They send you a box of pickles every month. It's just magical. And you currently have it so that every month you go into your bank account and you pay the bill, right? It's kind of annoying, but it doesn't take that long to do. It's really pretty fast. It would be better for you if you had the automatic bill pay for it, right? But to do that, you'd have to like set it up, right? That's why you don't. So, if currently you're at some sort of unstable thing, right, like, or high, it's uncomfortable, it's somewhat annoying to pay your bill every month. You'd be way more stable and calm if you just automatically did it, but it's challenging to get that thing set up. Yeah? What enzymes are going to do is they're not going to mess around with where the reactions start. They don't, the reactions already exist. They also don't mess around with the end product. This overall change in G, they can't touch that, okay? What they can touch is they can mess around with the activation energy. So they can make this activation energy, you can make it so you need less. Concerns about this? I'm going to repeat myself about 18 times between now and 11. So enzymes speed these things up just because it makes that bump that the reaction has to get over, it makes it lower. If you set up two friends and you know this person would be great with this person. Neither of them know each other. They would have great chemistry if they got together, right? You can't change how they are originally, and you don't change their chemistry as a couple, but you can get them together faster than it would have happened otherwise. Yeah? Enzymes are going to do the same thing. They're not going to change the original thing, they're not going to change the final thing, but they're going to make it happen in a biologically meaningful time frame. Okay? Here's this picture. I have a stick. You can see up there on the black line, so we see on the y-axis that free energy, right, starts out high. At the very end of everything, you end up with lower products, right? Great spontaneous process. We've got delta G, beautiful. Without the enzyme, the free energy bump that is called EA, it's activation energy, so it's written as energy of activation. It's really high. Takes up a lot. This isn't going to happen unless like the whole system's really hot or something like that. Without an enzyme, with an enzyme, it lowers that, makes it more able to happen. Products again are the same, but of course the reaction there in red, it's easier to go. Yeah? Great. So, words, more words. This class has become a vocabulary class, I realize, but you all are doing great. You have the enzyme, you have the thing that the enzyme acts on. The thing the enzyme's acting on, that's the substrate. Okay? Substrate, enzyme. When they're together, that's called an enzyme-substrate complex. Alright? And the reaction that happens is specific, meaning the enzyme can't just act on everything. It has a or a few substrates that actually fit. Okay? We will see why in a minute. We'll see why now. It's because the spot where the substrate fits into the enzyme has a specific shape. So some things fit and other things don't. The spot where that substrate fits is called the active site. So if you were thinking of like a door, where the key fits is that keyhole, right? It's a very specific shape. Not every single key in the world can fit into your keyhole. You need the key that's the right shape to fit in. Yeah? Induced fit is something enzymes can do. It's kind of clever. It's the substrate, as the substrate comes in, the enzyme and substrate can kind of like squiggle to fit just right. You can like jostle into position. I have video. You'll see it. It's great. Here's an enzyme here. Big protein, right? This big blue thing, right? The active site's right here. This substrate can fit in beautifully. And as it fits in, this enzyme kind of like folds in around it. So far, so good? This is going to be really great for this the molecule doesn't care. But you want a reaction to happen with this molecule. As this enzyme circles around it, things can happen. First of all, what should we call this enzyme? Like, pick just a given name, because I want to call it something rather than just enzyme. Suggestions?
Green.
Say it? Green Lantern? Okay. Green Lantern, our little Green Lantern enzyme right here. That's what you said, right?
Yeah.
Yeah, beautiful. It's hanging on right here. We've got the molecule there in the middle. This thing happening, as Green Lantern circles around its substrate, it can make a reaction happen that wouldn't happen otherwise. So maybe you've got two substrates that are going to come together and you're going to like switch the bonds around. It can put them so that they're facing the right way for this to happen, right? I said I wouldn't use my kids as examples, but I'm going to. 2-year-old, fantastic. She's a wonderful child. I love her dearly. She's in the phase of life that like she needs to do things herself. It's wonderful. And takes forever. And she's convinced she has to be the one to clip her car seat buckle together, right? She can do it. It takes so long. Just left to our own devices, it takes a very, very long time, right? Me, because we need to have this happen in a biologically meaningful time frame, I can help her get the little buckles situated right next to each other, the right orientation, and then she can do it. Click, right? Enzymes are going to do the same thing. Like, okay, this has got to happen. Yes, you little substrates, yes, you do your thing, but I'm going to help you be in the right spot to make this happen. Right? We're good with that? It can also, if you're trying to break something apart, the enzyme can like strain those covalent bonds, right? So the covalent bond needs to break anyways, but the enzyme kind of pulls a little bit. It can make it so that there's some little snuggly environment for it. So maybe this reaction would happen better if it was slightly acidic or slightly basic, or maybe the reason this little molecule isn't breaking up but it want it to is because it's got a whole bunch of hydrophobic regions that are like squished in this inside. So if we can exclude water, it can have a chance to undo itself. Yeah, we're good with that. It can bond with the substrate. It can stabilize things. It can do a lot to make this feel happy and calm. We're good? And that's all this. So active site's going to lower the activation energy. I'm going to ask that, like, what do enzymes do? Probably on next week's quiz, probably on exams, probably all the time. Enzymes, lower activation energy, right? Don't change the overall delta G, but they do make these things go faster. So here's a picture. We've got little substrates, high substrates. They go into the enzyme, right? They're held there. Oh, two of them. Okay. Substrates are going to get changed into the product. The product's just the end result. Products get released, and now they've cleared out the enzyme. This enzyme, okay, so at the beginning we have substrates. At the end we have products, right? We used up our substrates and we made products, right? Did we use up our enzyme? No. Did we make a new enzyme? Nope. We had one enzyme going in. We have one enzyme going out. This enzyme can catalyze another reaction. It can just over and over and over. Yeah? Beautiful. So, enzymes are proteins, right? Okay, good, yeah. Lots, excellent. Remember how proteins have a specific shape? They have to be folded to do their thing? And different environmental conditions can change the shape of a protein, right? So like, when you cook egg yolks, they change shape, right? Okay. Enzymes are proteins, they have a specific shape, and they've got to be a specific shape for their reaction to happen, right? To be able to bind this appropriately. So they work best in specific environments. I need to show you some pictures. Most human enzymes, you've got tons, loads of them. Most human enzymes work really well at 37 degrees C. So what we're looking at this chart is we've got temperature in C down here. Up here on the y-axis, we're seeing just the rate of the reaction, how fast this thing happens. Okay? So in black, here we have the reaction of some typical human enzyme, whatever it is. If it's freezing, if it's at 0, right? This reaction is just not happening. Okay? The rate of reaction is very, very slow. As it gets warmer, it happens more quickly. When it's at 37 degrees C, the enzyme is just in the perfect shape to just have this thing happen a bunch, right? And then as it gets hotter and hotter, as it goes beyond 37 degrees C, these specific enzymes function less and less well, right? It's not that just the hotter it is, the faster it works, right? There's a specific window there for it. Yeah? And there's not something magical about 37 degrees C that like all enzymes work. It's just human enzymes happen to work at about 37 degrees C. Any guess what human body temperature is? Yeah, so yeah, that's in Fahrenheit. Any guess what is in C? Yeah, 37. Right? You okay with that? human enzymes function in the human body, and they function at human body temperature. So one of the reasons why fevers are dangerous is if body temperature changes dramatically, you can make it so that the enzymes don't function appropriately, right? Because you've gotten too hot, okay? However, it's not that these are the only enzymes that exist on the planet, right? So there's some little bacteria buddies that like hang around in sulfur hot springs. They love it. They do great. They live in really, really hot environments. They live in environments that are about 80 degrees C. They're really hot. And their enzymes, we're looking at here in red, at 37 degrees C, they're not even like, they haven't even turned on really. Their rate of reactions work beautifully at here about 77, right? They function at different temperatures. And it's not that, and enzymes can do other things too. So, for instance, we're looking at a similar chart, rate of reaction. This time we're not looking at temperature, we're looking at pH. Remind me, low pH, does that mean acidic or basic? Acidic. Beautiful, it's acidic. Okay, so pepsin is this enzyme that lives in your stomach. It does its thing. Note how if it's absolutely super acidic, it's 0 pH, the rate of reaction is pretty low. But at about a pH of about 2, which is about where your stomach's at, this thing just works beautifully, right? But as it gets more acidic, it doesn't work so well, right? Because of where the hydrogens are, like it just doesn't, the shape isn't right. Okay, so that's pepsin, that lives in your stomach. Your intestines aren't acidic like your stomach is. Your intestines are slightly basic. Okay? You've got trypsin, it's another enzyme, it's supposed to do its job. It works at a pH of about 8. Alright? Because just the way it's shaped, it needs that pH to function. Yeah? This coming week in lab, you're going to be playing around with this, being like, okay, what happens if I get my enzyme hot? Or cold? Or throw acid on it? Like, how does it work? Questions about this? Are we comfortable with how to read this chart? Yeah? Okay. Deep breath, we're still in enzymes, but we're on to a new topic with enzymes. Everyone feeling okay with the transition? Our little enzyme buddy back here, I guess we would call it Green Lantern, right? Beautiful little enzyme. We want it to work beautifully. It works best if it's in the right shape. It works best at a certain temperature, certain pH, certain conditions, right? There are certain things that can help it. They're called cofactors. They're not proteins, but they help our enzyme out. Just little helper buddies. They can be something like metal ions are going to do this. They can be organic. So it could be something like magnesium. It can be something like vitamin D. Yeah. Most vitamins act as coenzymes, right? They're not the enzyme itself, but they help enzymes do their job better. So far, so good? You have things that can help enzymes. You've got environmental conditions that can hurt enzymes. Could you imagine some sort of chunk of something that could hurt an enzyme or inhibit its ability to function? Yeah? Right? Those are called inhibitors. There are two types of inhibitors. I mean, there are thousands and millions of possible types of inhibitors, but they come in two versions. Competitive inhibitors bind at the active site and stop it from doing its thing. So, back to our key and lock example. You've got this lock, this key fits in beautifully, right? If you chew some bubble gum and jam it down the keyhole, have you made problems? Yeah. The spot that the key's supposed to fit now doesn't fit right. okay, because there's something else there. Yeah? Um, that's called competitive inhibition, because it's competing for the same active site, right? Um, non-competitive inhibition isn't competing at the active site directly. It's not like something's there at the active site, but it's binding to the other part of the enzyme and changing the whole thing's shape and making it so the active site doesn't work so well. All right? A lot of toxins are enzyme inhibitors. Like cyanide is an enzyme inhibitor, and it's very toxic because it stops the enzyme from doing what it's supposed to do. Yeah? Here's the picture for this. Oh, yeah. Da-da-da. Here's the picture for this. Normal binding here. The substrate hangs out with the active site. The reaction happens. It's great. Competitive inhibition, this other thing that doesn't even really the enzyme doesn't do anything with this. It just sits there. This whatever molecule this is just plops itself on. The substrate can't bind because its active site is clogged up. Yeah? That's competitive inhibition. It's fighting for the same spot. Non-competitive inhibition, nothing's blocking the active site, but something stuck to the enzyme someplace else, and now the whole thing's in a different shape. Right? And now, this substrate can't fit, the active site isn't as good as it used to be. Yeah? If you want to take an example, if you're studying for biology, which you should be, right? Dedicatedly studying for biology, if there's something that's competing for your time, so say you're studying for physics, right? That's a competitive inhibitor of your biology study, right? Because same thing, but you just now have your time someplace else, right? If the time hasn't been hurt, but you just like feel like crap because you don't feel good, it's a non-competitive inhibitor. It's not directly competing with your time, but it's directly stopping your ability to have this happen. We're okay with the distinctions here? In lab next week, you're going to look at competitive versus non-competitive inhibition. And the kind of interesting thing is, if you've got non-competitive inhibitors, and if you have enough of them in there, you just shut your reaction down, right? Because you have non-competitive inhibitors, the substrate comes in, but all the enzymes are more or less broken, or at least inactivated, right? With competitive inhibition, you can solve competitive inhibition, more or less, by just dumping a ton of substrate on it, because then every time this enzyme is looking for something, it always finds the substrate rather than the inhibitor, because there's just so much more of the substrate. Side note, are you guys interested with like random side notes? Okay. I try to stay focused, but then I get excited. You all know better than to drink antifreeze, right? Don't drink antifreeze. It's very bad. Sometimes dogs will drink it because it tastes sweet, but it's very poisonous. There's an enzyme that's supposed to be doing something. I can't remember what it's supposed to do. It's supposed to be doing something. But antifreeze acts as a competitive inhibitor, and it like just plops itself on and sits there, and doesn't come off. And so you basically bust your enzymes, and you can die, right? The part of this that I think is interesting is the way it's treated, and like don't do it yourself, okay? Like if your dog or somebody you know drinks antifreeze, get appropriate medical attention. Don't be like, I heard in my biology class, okay? Just the treatment for this is, I'm not making this up, it's just to have the person or dog drink a bunch of alcohol, like a lot, because that's not like, well, enjoy your last few days. It's because alcohol also binds to the same enzyme, and so what the plan is like, okay, crap, the antifreeze is going to come and hit the enzyme and stick, and that's a problem, and it's going to break your enzyme, and bad things will happen. So what we're going to do is we're going to pour so much alcohol into the system that every time the enzyme looks for something, it doesn't see antifreeze. It sees alcohol instead. And alcohol just like sits there and like just chills there. And we need this for like 12 hours until the antifreeze can pass through the system. The person will be very drunk, but that's okay. We're going to pass the antifreeze through the system. Alcohol doesn't stay forever on the enzyme. It gets pulled off. So then it'll be okay. I know, right? Can you imagine that though? You show up, you're like, oh no! My husband accidentally, drank antifreeze. And they're like, do you have any alcohol? Right? But it does work. But the point of that competitive, non-competitive inhibition, yeah, okay. I try to stay focused, people. I do. I mean, I don't try hard, but I do try, sort of. Okay. You've got to control these enzymes, because otherwise, everything is a mess. Like, the cell would just be a mush of a mess. So, you have all these enzymes that can do tons and tons of different things. So, you tightly control what things they're doing, right? So, having an enzyme turned off isn't always bad. Sometimes you want it turned off. Sometimes you want it turned on. You can control enzymes in two main ways. You can either control the genes that are going to be responsible for producing the enzyme, but that's a different story. So you should be like, we want to make a lot of this, or don't make that, like, we don't need that. You can also regulate the activity of the enzyme itself. You can put something on it that makes it go better. You can put something on it that makes it not go so well, or turn off, right? So you have all these enzymes can do a million different things, and you regulate their activity. I know I'm going fast. Is this too fast? People doing okay? I'd like to get you out a little bit early so you have time to do your quiz, or have time to at least think crabby thoughts about Almet. Apparently, I think they're fixing it.
They are fixing it, but it hasn't made them to know if it's
Oh, okay, cool. Well, anyway, we'll see how it goes. Okay. How you can regulate enzymes? It's called allosteric, which just means related to shape. You can stimulate or inhibit this enzyme. So, what this means is lots of things. What this means is enzymes are going to, a lot of these notes I put in just so you can study from them. I like talking about pictures better. Enzymes have this active site, right? And the active site needs to be in a certain shape for the substrate to come and attach properly, right? But enzymes are proteins and they can be in different shapes, right? So most enzymes should have an active state that the substrate can fit, and an inactive shape that doesn't fit quite right. So an allosteric activator, something that's going to encourage that, is going to be something like this little green blob here that stabilizes this enzyme to hold the active site into the on position, right? There's a shape that works really well for the substrate. It's going to help hold it on. You know how with a push lawnmower, when you make it go, you need to pull the little handle back? If you tape that on, right, right, you can help hold it in the on position. There's also going to be a non-functional, non-active, so it's called the inactive form. An inhibitor is going to help hold it in that off position. So whatever's trying to come can't fit because it's being held in the off position. Yeah? Another version of activation is called cooperativity. So you've got this one over here that's not on the active site itself. This one, as one substrate binds, it makes it easier for other substrates to bind too. That's actually fairly common. Questions about this? It's in the same general category as, yeah, you can encourage the enzyme's ability or you can stop its ability. And both of it, it's not like one is bad and one is good. You need both of them and you want them under metabolic control. Yeah. Okay. Does anybody here feel like most rooms are either too hot or too cold? Which way? Too hot, too cold? Too cold? Too cold? Okay. Yeah. the room's always too cold, right? Do you have control in your dorm room over the thermostat? Yeah, you're like, this needs to be 85 degrees, right? Woo! Not 85, 75, 72, okay, 72, 62. It's colder than 62 in your dorm rooms?
No, it's 62 in my
Okay, good. Whew, nice, okay.
I keep the coldest.
You feel like, make this be beautiful. Okay. Yeah. Whew. And it seems often, um, I yeah, it seems often that people who have very different set points for how they want temperature end up being roommates. One roommate wanders past and is always turning the thermostat up, and the other one wanders past and is turning it down. Okay, yeah. This half the class is nodding deeply. Are you two roommates? No, okay, I feel like, yeah. Okay, so let's say you feel like you want it to be warm, right? Okay. So what you can do is you're gonna go to your thermostat and be like, you know what? Sure, 72 degrees. I want it to be 72 degrees. Or, I know, let's have it be 85 degrees. I want it to be 85 degrees. Turn that thing up. What the thermostat is going to do in response to this, right? It's gonna notice the temperature that it is around. And be like, oh, send a signal to make it warmer, right? Signal's gonna be produce heat. Heat gets produced, right? And that heat that's produced comes back and shuts off the thermostat, right? Thermostat, now the thing that it made turns itself off, right? If it didn't, if it just kept cranking out heat, it would get hotter and hotter and hotter and hotter, and you make more heat than you want it to, right? You want the thing that gets made to shut off the go signal. Is that fair? Um, in a cell, this is called feedback inhibition. So the thing that you make feeds back onto the metabolic pathway and inhibits that pathway, shuts down the pathway. This is really good because otherwise you make way too much of whatever it is you wanted, right? You want this particular sugar, but you have to break down something to do it, or whatever. You don't want to just run this forever, right? You want to make however much you need and then be done, right? Here's a beautiful pathway. So if one of your cells doesn't have much isoleucine, this is a problem. You want to make isoleucine. You like this is your life's goal now, right? You can get done. There's this enzyme here that does it. It takes threonine. It grabs this molecule, has but I've conveniently glossed over as several intermediate steps. It does several things to it, and ends up with isoleucine. Great, right? So if you have no isoleucine, this enzyme right here grabs threonine and just change, several metabolic steps. But eventually you end up here, right? What the beautiful organization of metabolic pathways. These things can have like 70, 80, 90 steps. They're so cool. And they're long. But this end product acts over here as an inhibitor to this first enzyme in this step, in this process, right? So if you have isoleucine, it binds here, and it stops this enzyme from going to the gusto. Do you see that? This right here is a non-competitive inhibitor, right? So if you have a bunch of isoleucine, some of that isoleucine stops the enzyme from functioning. And that's good, right? Because now you have stuff, you don't need it, right? As isoleucine gets used up by the cell, right? As the cell using it, doing whatever it does with it, you then free the enzyme. And so now you don't have much isoleucine in the cell, the enzyme's ready to go, and it's going to turn off. Do we see that? Feedback inhibition process. It's very clever. Questions? Great. And the actual steps can be different, and different metabolic pathways have different numbers of steps. And you can have lots of different kinds of inhibition, but that's the basic idea. Finally, okay, well, like 3 final things. One final slide, a video to show you, and then those little clicker things. The last thing is enzymes are put in different places in the cell. So there are enzymes all over the cell. But say enzymes for cellular respiration, a bunch of them are crammed into mitochondria. Enzymes for the things that are important on the membrane, you cram in enzymes into the membrane, right? Like, you put them where you need them as a cell. And it just is very helpful. Okay, I have video. You don't need to memorize. Oh, so online people, I'm going to stop recording. Do watch this video. You don't need to memorize all the different things. They're going to talk about different names of stuff. But the idea is to see what's happening here. It's going to show how the enzyme's getting this done. So I'm going to stop recording. Online people, do watch that.